Hello everyone, my name is Ashfaq Ahmed and I work in the field of structural biology and bioinformatics. You are watching Bioinformatics Insights. Today I'm going to cover a very important video about understanding matrices of the PDB structure. Most of you people are working with PDB structures taken from PDB for your drug discovery or drug designing or some other research. So you should know which structure to select and which structure not to select or to filter out. So the and we call those things those features matrices. So what actually is PDB? If you don't know, so there is a short description that it is a global repository or a global database. It's a uniform database which stores 3D structures of macromolecules like nucleic acid, proteins, and complexes of proteins with ligands and other things. Okay, and it is managed by the Worldwide Protein Data Bank and these organizations. It was established in 1971, and so far, almost this amount of experimentally determined structures are present, and greater than 1 million CSMs are present. CSM stands for Computed Structure Models, those models which are computed by the AlphaFold or some other methods, but not experimentally determined. So we call them CSMs. So take care when you are taking a structure from the PDB. There is a short headlines method. So the method will tell you whether it's experimentally determined or it's from the CSM or computationally determined. Okay. So just I want to remind you once again that PDB contains the structures from three experimental methods. One is NMR, another one is X-ray crystallography, and the third one is cryo-EM or cryo-electron microscopy. So apart from these three methods, the all other structures are CSMs or computed structure models. Now, this is a screenshot I took from the PDB database for the PDB identifier 4EXS. So you can see a DOI and a classification of the protein or the enzyme, the organism from which the protein belongs, and the expression system in which expression system this particular protein was expressed, and does this protein contains mutations. One of the problem is that sometime you are selecting your structure but you do not care for this. So this is must just care for mutations. Like if you are interested in a raw structure or in a wild type structure, but if by mistake you select or you download a structure with a mutation and you came to know later on, then it will be a difficult time for you to repeat all those experiments again and again. So it's better to understand this thing that whether the structure you are downloading or you are interested to download, is it contain any mutation? If yes, so what are those mutations? And the structure deposition date, I think that that is okay. And who deposited the structure, so that is also not important. Okay, now there is the detail. You can see the method. So this one is X-ray diffraction method. As I told you in my previous slide that there are three methods: diffraction, NMR, and cryo-EM. So it's experimentally determined one. Okay, now here is the resolution and the R value. The R value are classified into work and observed, but the most important one is free. So what it stands for, and also in the final slide I will discuss that, like the quality of the structure if you are selecting it for a ligand binding experiments or docking or all other those things. Okay, so these are the matrices we will discuss quickly. What are the importance of these matrices? Just three important I coined for you on this slide. First of all, these matrices provides you the quality and the reliability of the protein structures. The structure you are using, is it a quality structure or not? It is must. It is must for you to know and to mention it in your thesis and manuscripts. And it's a reliable for experiment to be used or not. You should know this. Okay. Option two, these matrices also provide some insights for the accuracy of atomic coordinates and the agreement between the model and the experimental data. What does it mean, the model and the experimental data? Let me give you a brief explanation. When a crystallographer makes protein crystals, so they bring it to the synchrotron, and where it is shooted by a sophisticated machinery. We call it X-ray diffraction or X-ray XRD. So it is shooted by the XRD machine or the X-rays, X-ray rays. Okay, then the synchrotron people will provide them some data. The data will be the data of electron density. We call it electron density map. So the crystallographer going back from the synchrotron facility, they will have the data of electron density map. Then they keep those electron density map in front of themselves, and they try to solve the structure of each and every atom present in that protein. So in some cases, if there is a noise in the data, then the crystallographer remains unable to resolve all parts of the structure. For example, if the structure has two domains, so maybe the first domain, the data from the electron cloud is very clear, then easily they can spot out each and every atom of the protein of the domain 1 from that data. But there is a possibility that due to some experimental noise, due to the data shooting, uh, the second domain contains the data from the second domain, the electron density, may be not very much clear. So in that case, you often see some missing residues of the loops and other regions. So I don't want to go into bigger detail, but the fluctuating regions are almost a bit difficult to capture by successfully on the electron density map. And these loops and these things, they are with high B factor, or we call it beta factor or temperature factor. So that's why they are a bit on the higher side. So you should know before selecting a protein that what is how about these matrices, because these matrices will tell you the accuracy and the ability of the structure to be used in your research experiment. So the third one is understanding these metrics is important for your research if you are particularly in if you are using in the drug discovery and molecular biology and structural bioinformatics. This is particularly important. So this video may be theoretical for you, but I rank this video very high because it laid the foundation of your research, because you are selecting a protein structure in the start. Okay, let's come to the business. So the first one, you will see a method, but I explained the method part. Now the second thing, you will observe resolution. What actually is resolution? Resolution is typically measured in angstrom. For example, if you are seeing 1, so that means 1 angstrom, 2 angstrom, 3 angstrom, and so on. What actually it explains? It explains the details in a 3D structure derived from the experimental data. What is experimental data? I explained in my previous slide that is actually the data of electron density maps received from the XRD experiment, which shooted the protein crystals. Okay, so the structure derived from the gap between, or the difference between the structure and the experimental data. If this gap, or is this difference is very high, then in that case the R factor will be very high. Sorry, the resolution will be very high. Okay, let's say high in number, I mean. Okay, so there are low resolution values, just like 2.0 angstrom or less. Okay, by number I call it low resolution values. Okay, but it indicates the higher quality. Remember, what does it mean, the 2 and 3? What is the difference? If a structure has a 2.0 angstrom resolution, and if a structure has 3.0 angstrom is a resolution, what will be the difference? The from 2.0, it means that in this structure provides you the atomic details up to 2 angstroms. But this structure provides you the atomic details up to 3 angstroms. If you are interested to see the atomic details are at the level of 2 angstrom, this structure will fade. So the lower resolution value like 1, 1.5, 2, it will be considered a higher quality. And in structure biology, we will call it higher resolution. You got my point? If the value is lower, so the structure is of a higher quality, and we will call it higher resolution structure. But if this value is higher, like 3, then in that case, if we compare these two, then this one will be considered a lower quality, or with less atomic details, or a lower resolution. So for example, if I ask you, you have these two structures. If I ask you which one is a higher resolution, so your answer should be 2, because I mean from, not by the number, I mean by the quality. So the quality of this structure will be better. And if you have 1.5 resolution, then that one will be the best from these two. And this is the concept, like for example, I explained like 2 angstrom or less means the structure provide clarity up to this level. Okay, a structure determined at 1.5 angstrom resolution would be considered high resolution, while one at 4 resolution would be considered low resolution. I hope if you have more than one structure for a protein from now on in the PDB database, so you should be able to select the one with the best resolution. I mean, with resolution value lower. What is R value? And we also call it R factor. It is actually the measure of the experimental, the difference between the experimental data and the structure which was calculated from that data. So if that difference, if that data is on the higher side, for example, if the spots on the electron density maps shows something else, but due to some reason the crystallographer managed to derive a structure from that data, then the computational program matches the difference between that experimental data and the structure. If this difference is on the higher side, the R value or the R factor will be higher, and that's why it will compromise the structure quality. So in short, the lower R value or the lower R factor is the best or is the better one. So the second thing you have to take care of the R value or the or factor. Okay, so a lower R value indicates a better fit. I explained to you why, suggesting the model accurately represent the observed electron density. or other experimental data. A higher R value indicates a poor fit, and it indicates potential model error. So if you have the choice to select a structure from the PDB, because most of the time, if you are selecting a structure from the PDB, so most of the time a PDB contains more than one structure for that protein. So there are certain suggestions I would provide in this slide, like which one you will select. First of all, go for the one that covers majority of the sequence area. For example, if one structure is for 10 amino acid and the other structure is for 30 amino acid, so I think the one with 30 amino acid is far better. But the second thing is you should know whether it is experimentally resolved, so you should check the method section. The third thing, check for the resolution. If it's 2 or below 2, so I think that that's better and you can safely select it. And finally, you should also take care for the R value. If the R value is very bigger, then that means the structure has a poor quality once again. So do not try to select or that structure or go for some another option. Okay. This is the concept difference between the experimental data and real structure, the minimum, the best. An R value of 0.2, that means 20% is generally considered good, while the R value zero point 0.4 or 40% or above is considered a poor structure. Okay. Now, finally, we will discuss the ligand structure and quality assessment. Uh, its suitability for drug discovery research. So I will perform this just for you quickly in 30 seconds practically. So I have a structure opened here. This is the structure of EG3UG5, and it is, if I'm not wrong, crystal structure of the arabinose, whatever, I don't know. Okay. Uh, Thermotoga, whatever. Okay. So we discuss majority of the things here. Now, can you see the ligand structure quality assessment? Yes. So now, visually, the if this thing is in this blue zone, it's better. Okay, it's good. But to clarify further, just click here. When click on this line, possibly it's a new thing for you. Wait, it's clickable. Come on. Okay, now when you click here, you will have this window. Now you can see it just the geometry and the bitter and the wars. So it's towards the bitter thing, the bitter blue part. But come here. Now it just tells you for the fitness for the ligand 85%, ranking by geometry, this model completeness 100%. So it's better if you are interested, you can check this thing also. And it's also another structure for from the same for possibly a homolog. You can check it here as well. You can click that, you can click there. So it's just a general indication for you. And finally, it's not the scope of this video, but let me tell you, this is the paper of the structure you are seeing or you are interested for your research. So whatever you do, you need to study this paper. This is on the PubMed, so you can click here, open here, and it's better to first study this paper and what is inside the structure, how many sequence lengths, how many chain, A, B, C, D, E. This is the image. And is there any ligand? So yes, there are two ligand, XYP and TRS. These are the structure. I think the TRS came from the experimental details, but most of the time you will see here just like chloride or just like glycerol. So they came from the normally they came from the experimental setup. They are not real ligands, and that's it. So if you're liking my video, so just thumbs up, and thanks for watching. Today it was a completely a kind of a theoretical video, but I thought it was necessary for those who are working with PDB structures and they don't have much breadth of the PDB structure. So I hope this video will help them a lot. Thank you so much. Share this channel and subscribe this channel. Thank you. Bye bye.